Thank you for listening to American Medicine Today. I'm Kimberly Brumell, joined by Ethan Euchre.
Glad to be here.
Junior Mint Jeff Wagstaff.
Thanks for having me, Kimberly.
Absolutely. And world-renowned orthopedic surgeon, Dr. Alfred Bonatti of the Bonatti Spine Institute.
Thank you for allowing me to stay here.
Yes. Always.
Your name's on the show, Doc. You got to be here.
Now, after decades of drug testing, mostly on men, a new device called the EVATAR may help tailor treatments for women as well. So joining us right now is Dr. Teresa Woodruff, professor of obstetrics and gynecology at Northwestern University and one of the co-authors of the study. So thank you for joining us, Dr. Woodruff.
Thank you. Happy to be here.
I'm torn on this one. I find it fascinating yet creepy, so I do apologize.
Why do you got to apologize?
But you've pretty much created
a reproductive system.
Yes, in a petri dish.
Yeah.
Well, really, it's not a petri dish. That's the key. A lot of people think of petri dishes because that's what we were all used to doing in biology labs in high school. What we've done is we've thrown the petri dish as far away from the laboratory as we possibly can because that doesn't really recreate the biology that we have in each of us. Namely, what that means is that we have a group of organs that all function together with the vasculature, the arteries, the capillaries, and the veins that move nutrients between organs. They communicate with each other, and then metabolic waste is eliminated. The petri dish doesn't allow for any of that. It's basically a single set of cells flat on a piece of plastic, and that doesn't represent any cell in any of our biology. The cells are basically sitting in their own metabolic waste in an incubator. We've really revolutionized that kind of technology by adding microfluidics, so fluid flow between different wells where each of our tissues are placed. We started with the reproductive tract because that really represents one of the fundamental systems that has to communicate between each other with very specific outcomes. So the ovary makes hormones like estrogen and progesterone, and the fallopian tubes respond by changing the ciliary movement to allow the oocyte to move down the tube to be fertilized. The uterus and the cervix change in their dimensions, and cells slough off as they go through a normal cycle. So this was a great opportunity to test whether or not microfluidics could actually support organ-level communication between tissues. When we started, we didn't know if we could really do this. We didn't know if we could get tissues to last for 28 days, which is what we did. We didn't know if we could get them to communicate with each other. We needed a universal media, which is what our blood does for each of us. And we didn't know if the function would be supported. I don't think there's anything creepy. I think it's revolutionary technology that supports breakthrough biology, not only for reproductive biology, but for all of biology. And I want to also say that all of this was funded by the National Institutes of Health, which is important for all of us to understand how these new tools that are going to provide personalized options for each of us in the future is being funded.
I understand why it would be important because, we're talking about in the late 50s, things weren't really tested on women at all, predominantly men. And all of a sudden, they prescribed a drug to pregnant women, which turned out to cause severe birth defects. And that's because they hadn't tested it on women.
Right. They didn't know what the effects would be on a pregnant woman because they only tested the drug on men.
Are you saying that they won't ever have to test on females with the type of advancements you're making here?
No. I think what we have is a new tool in our kit that brings us from the bench to people with tools that are more like how our own organs function. So we'll still use cells in petri dishes, but imagine now for cardiovascular disease where there are different phenomenologies that impact males and females. Bone health, for example. We've never been able to study bone or cartilage in the context of the rising and falling hormones of the female, estrogen and progesterone, nor with males. So we're also creating systems that provide the morning to night cycle for male hormone, testosterone, and then the outcomes that we would see on other tissues. This is really an exciting new way of thinking about the step between the individual cell in a petri dish leading us towards animal or human biology. I think it's going to hasten drug development in a really significant way.
Will just taking organs in that one particular part of the body, though, be indicative of how it will be in a human?
It's one more step toward that because we've never had integration of organs ever. We have individual cells within individual wells of a petri dish. We've never connected those tissues, so that's how this technology will allow us to move forward. So, for example, we can now put in bone or cartilage or adipose or muscle tissues and begin to look at the biology of drugs or their effect on those tissues during a normal cycle. All of us have cycling hormones, males and females, females, females a 28-day cycle, males a 1-day cycle. All of our biology really is informed by the underlying sex hormones, XX or XY, in each of our cells, and there's an imposition of these hormones. Now we're going to be able to study that biology. In the absence of studying hormones or sex as a biological variable, our cells have no hormones of either male or female. So at some level it might be pseudo-postmenopausal state, but not really. So we're going to amp up our ability as basic scientists to be able to understand more about the cell signaling pathways that are occurring in each of our organs using this kind of system.
You've been working on just the female reproductive system. I say just like that's some minor thing, but eventually, don't you think you'll be able to produce these different systems that could create essentially an entire human being? So whatever part of the body you wanted to test drugs on, you could. And here's what I was thinking. Would that replace animal testing, and would it speed up the FDA approval process because you have an apparatus that can accurately reflect how the human body would respond to these drugs down the road, and how far away are we from something like that?
The hope is that we have representations of our organs so that we could use iPS cells from an individual. And in our system, we have a liver to provide the opportunity for metabolism, and each of us metabolize drugs very differently. Being able to look at metabolism on a personalized level is going to really allow each drug to be tailored to each of us. And I think that's really important. We also have islet cells that we've been using in the system, and we can actually take the insulin out of the media so that the islets, the pancreas, is producing the insulin necessary for the other tissues. And we're studying male cells as well. Our hope is to eventually limit the number of animals in biomedical research and make it easier and faster and safer for these drugs to be tailored for our individuals rather than population, which is the way drugs get tested now.
Are you guys also going to do like a miniature brain? Because doesn't the brain give emotion to what you're doing?
Would it be conscious?
Not only that, but when you take drugs or different things, wouldn't the brain also have an effect on the outcome?
We're creating models for each of the organs. So we're allowing for the normal, ordinary function of each tissue to be mimicked in the system. And the best way this system is used is through communication between organs. So the endocrine system and the nervous system are the two great communication systems of our body. And the endocrine system has hormones that communicate information between all of our tissues. And that's something that we simply can't model. This system is going to allow for that ordinary communication that is relayed between each of our different tissues to be part of our basic science analysis of signaling pathways, to allow us to study drug interaction, to identify new drugs and how they might work, to begin to use each of the tissues as it is in our body. They are communicating together, they're informing each other, they're informing the biology of downstream effects of each of the drugs. That's what's really exciting about this new technology, is it's going to enable a new kind of biology that is going to, I think, open up a level of discovery that we haven't had in the past.
Well, we truly appreciate you coming on to discuss this. Dr. Teresa Woodruff, professor of obstetrics and gynecology at Northwestern University and one of the co-authors of the study. Thank you so much.
Thanks a lot.
Have a good one, Dr.
Take care.
Woodruff. Crazy cool science, sci-fi type stuff going on there. I love it.